now online. I hope the technique can provide us with access. Yes, it's Sander van Kasteren from Leiden University. We are very happy that you could join us here online and to provide you your lecture on in vivo click chemistry again, and now here with a closer application or a closer focus on novel tools for immunology. We are very glad to have you here, and the screen is now yours.
Thank you. I'm really glad to at least digitally be there. Would have loved to have been there in person and seen all the other talks, but teaching prevented me from joining. But as it's a short talk, without further ado, I will dive into it. And I'm also very grateful to Dr. Mikkola for providing a lot of introduction on the chemistry that I'm going to be talking about. And to discuss the topic or the urgency of the topic, I want to take you back to a paper from a few years ago where they essentially made patient-specific vaccines. So they sequenced the mutations in someone's melanoma and made peptides, a vaccine based on peptides, also on RNA, but that's a different one, based on the mutations of that patient to see if they could basically cure them. And the results were really rather spectacular. So these were no longer treatable patients, and they received a neoantigen vaccination, and 4 of them did not need any additional treatment to be disease-free for the duration of the study. So this was super exciting because essentially this vaccine activates cytotoxic killer T cells. So these are T cells capable of killing cancer cells in your body that had been deactivated during cancer's progression. So they designed essentially the peptides to perfectly bind MHC complexes for showing to these T cells and activating them. But then a closer look at the study actually revealed a major surprise that of all the peptides used in the entire study, only 16% activated the correct T cell. 10% activated helper T cells and killer T cells, and 50% only activated helper T cells and did not in any way activate the killer T cells that you need. And as we had optimized, or they had optimized, the peptides to bind only to the MHCs capable of activating these CD8 killer T cells, I was quite intrigued that clearly all the mechanisms of target selection that we have are insufficient, and there must be mechanisms at play that determine the antigenicity of a peptide in addition to its ability to bind an MHC. And we need to understand this better so that we can maybe start getting higher percentages of these peptides to work in these vaccines. So for the biology of this, I'll have to take you to this cell. It's called a dendritic cell. It's essentially the key activator of your immune response and of these killer T cells. So what happens during immune activation is that these cells take up an antigen here in gray through endocytosis and then route it to MHC1 complexes that are normally reserved for loading misfolded proteins or degraded proteins from the cytosol and the ER. So these are then presented on the cell surface where they can be sampled by T cells. So as you see from the number of arrows, and some of them actually contradict one another, we do not know how this routing happens. And another aspect that we do not know, and I won't go into the details of why this is important, but it is very important, is we don't know how fast all these processes occur. So what I thought, can we use click-to-release chemistry to study the timing and the routing of these antigens in living dendritic cells and eventually in vivo, so that we can find new parameters to improve CD8 or cytotoxic killer T cell activating vaccines. So I want to look at 2 projects, one involving timing and the other involving the routing of antigen, and we both use click-to-release chemistry. I don't have to introduce click chemistry, Nobel Prize last year. The part I interested in was essentially developed by Caroline Bertozzi, where she showed that 2 reagents in a living cell, or now in even a living mouse, can be ligated to form a product. Hannes already beautifully introduced the work from Marc Robillard and also Eric Meggers and others that show the inverse, that you can also take a protected product and break a bond in vivo, which is now even in human clinical trial at the moment. So my first foray in this actually used a very old-fashioned reaction, the Staudinger reduction from the 1920s, I believe. And what we did is we made a synthetic vaccine antigen in which the bit that is recognized by the killer T cell, the lysine side chain, is masked by an azide group. This peptide is then processed, routed through the cell, presented on the cell surface, and not recognized by a T cell clone that we have against this epitope. Then after doing chemistry on the cell surface, we can reduce this azide back to the cognate amine and control chemically the activation of a T cell. It works really quite cleanly. So this is T cell activation in concentration of the long peptide, of the wild type peptide, and of our azide modified peptide. So we can completely kill T cell recognition. And then upon addition of a reducing agent, a phosphine reducing agent, we can rescue the majority of the T cell response on the surface of the cell. Sadly, this compound at the concentrations that we would want to use it is toxic. Its in vivo characteristics are terrible, so we were a little stuck with this project. And then Marc Robillard released the transcyclooctene reaction. And we essentially did the same with a small trick that we loaded the minimal epitope modified with a transcyclooctene cage. And on the surface of an antigen presenting cell, we could activate this and do this with a variety of tetrazines, with a variety of release kinetics. Some of them release within minutes. The reaction seems to be quicker on the surface of the cell than in a dish, which is quite strange, but quite handy. And we also have tetrazines that can completely block the T cell activation. So the first story, which is actually an unpublished story, is thought, can we essentially do the same as for our Staudinger-based chemistry, but use a TCO-based antigen, let it be processed, and then on the surface, exclusively on the surface of the immune cell, activate the cell. Because in that way, we essentially have a clock that we can monitor how fast new antigen appears on the surface of a living dendritic cell and then eventually in vivo, because we've also tested the short peptide work in vivo, and that works. So the first thing we did was make the long peptide. So we used a slightly more water-soluble TCO variant that we stuck on a peptide that needs processing, pulse for a given time, then chase for different periods, release and add a T cell, or not release and add a T cell. We can also look at chase periods where after release, we can measure how long these things stay on the cell surface. So the normal immunology trick is that you, rather than deprotect, you fix. So here you see uncaging of the long peptide after fixing. So you see more and more peptides and more T cell reactivity occurring. And we can quite nicely mimic that by, rather than fixing, adding a tetrazine after these different chase periods that you see here. If we don't add a tetrazine, we don't activate our T cells, which is quite a nice clean system. So there's no hydrolysis of our TCO throughout the routing pathway. This we're now also using to study surface stability, and we're actually translating this in a mouse so that we essentially have an in vivo fixative for, just looking at the time, yeah, so that we have an in vivo variant of a fixative so that we can stop an antigen processing pathway in a mouse in real time, and the experiments of that are currently ongoing. What we've also done is we've tried to use a different tetrazine that you see here, one that's targeted to a specific organelle, to see if we can use click to release to learn something about the actual route that the antigen takes through a cell. So as I've shown here in this pathway, there are two competing schools of antigen cross-presentation, as this process is called. One believes that antigen escapes from a mild endosome that's not very acidic into the cytosol, where the proteasome does degradation, and by a variety of mechanisms, the peptides then reach the ER where they're loaded on MHC. There's another school that believes that antigen and MHC1 are co-internalized into an acidic phagosome. Endolysosomal proteases degrade the antigen, and peptides are loaded on MHC in this phagosome and presented on the cell surface. So I was quite curious by this because this vesicle is believed to be acidic and this one is not. So I thought, what if we take, to prove that a cross-presented antigen actually encounters an acidic vesicle, what if we take a caged antigen and have a tetrazine that accumulates in this acidic compartment? So essentially, we did the same processing of the long peptide to the short peptide and the release, but we used a tetrazine modified with a lysotracker modality. And what was really quite striking was that if we gave this compound, which is number 3, if we let it pre-incubate with the dendritic cells, wash it away so it accumulated in its lysosomal compartment or in its acidic compartments, and then gave a long peptide, we saw no T cell activation. And if we saw no T cell activation. And if we gave our peptide first, let it react, and then added the tetrazine to see if we could deprotect just anywhere on the cell surface, then we did see T cell activation. These experiments were included to show that short peptide, when you have intracellular tetrazine, so short peptide that only exchanges on the surface, is never activated either. So this is a very nice negative confirmation that this acidic compartment is either not very acidic or doesn't take part in the cross-presentation pathway. However, we needed a positive control as well. So, and my apologies to the student Mark de Geus, who spent quite a few months making these CD1 ligands, which is a glycolipid antigen used for the activation of other T cells, and he modified them with a TCO. And with these experiments, when we added the lysosomally targeted tetrazine and then added these reagents that we knew traveled via lysosome, we did get activation of the glycolipid-specific T cells. And 1 and 2 are 2 different variants of this parent molecule. So that already gets me to my conclusions, and that is that I think click-to-release chemistry based on TCOs and some of the more advanced variants that Mark Robillard and Hannes are both making is a very, very powerful tool for the study of kinetic processes in vivo and to study topological processes. So if by restricting them to organelles, and Milan Vrabel has, for example, made a mitochondrion-restricted tetrazine, you can actually study where in a cell something is happening in a live cell, essentially in real time. We think that cross-presentation does not occur via an acidic compartment, but therefore that the non-acidic compartments may play a more important role. And we're using this chemistry to make an in vivo compatible version, so essentially to be able to fix in an animal. And with that, I want to thank Joanna, who started this whole project, and Nina and Mark and Alexi, who are really the drivers of the engaging chemistry. And I thought, as you are now in Switzerland and probably a few of you ski, this is what we have to resort to in the Netherlands, and that's skiing inside a hall. And with that, I want to thank you for your attention, and I'm happy to take any questions.